Welcome, everyone. Thank you for being here. My name's Rich Lyons. I'm the chancellor here. Um, this event, I'll say more about it, but Berkeley's known the world over because of our research, and most specifically our fundamental research, and that comes from faculty excellence. We all know that. That is sort of the most fundamental of the assets that define Berkeley. And that's what we're here to celebrate today is our faculty excellence. For more than 110 years, our Faculty Senate has singled out two faculty members each year, as you know, who embody this excellence to deliver our Martin Meyerson Faculty Research Lecture. These distinguished professors have conducted research that has changed the trajectory of their disciplines and of our understanding of the subject they have dedicated their careers to. The lectures shine a light on an essential part of our mission, the creating new knowledge part of our mission. The curiosity and creativity that fuel the quest, our quest, collective quest to learn and understand, are at the heart of our commitment to making this world a better place. Through what we discover, through what we teach, and the public service we provide. This year's lectures represent the continuation of a treasured tradition that has recurred annually, with one exception. As I mentioned last time, in the wake of World War I and the influenza pandemic, the faculty research lectures were suspended. That was 1919, but that's the only exception. In 2020, when virtual events were in vogue and Zoom kept us together, these lectures went on. Being selected to deliver a faculty research lecture is indeed a high honor. To stand out among the thinkers, the peers who exemplify academic excellence, it's no small feat. For students, members of our campus community, and the public, this is a wonderful way to experience scholarly research of the very highest caliber. I'd like to welcome the past recipients who are with us today. Professors, please stand when I read your name. Now, last time I said to hold your applause until everyone has been recognized. Let's applaud after each person, okay? I think it's just more fun. I'm not going to try and control you. Chancellors shouldn't do that. Robert Alter is here. Thank you, Robert. Walter Alvarez is here. Thank you, Walter. Daniel Boyarin is here. Daniel. Henry Brady, who gave our last FRL. Thank you, Henry. Alexander Chorin is here today. Alexander, there we are. Marvin Cohen is with us today. Marvin. William Dietrich is here. There, William. Inez Fung is with us. Inez, yeah. Thank you. Catherine Gallagher. Robert Hass is with us today. Thank you, Robert. Martin J. is with us right here. Thank you, Martin. Thomas Lacour is here. Thank you, Thomas. Stephen Lindau. Stephen, here. Tony Long is with us. Tony Long. David Rowlett is here. Thank you, David. Barbara Romanovich is here. Thank you, Barbara. Pam Samuelson is with us. Pam, right there. Thank you, Pam. Randy Sheckman is here. Thanks, Randy. And Bob Tejan is here. Bob, thank you. Francesca Rockberg is here. Francesca, thank you. John Clark is here. John is here. I believe he's here. Yes, thank you, he is. And Birgitta Whaley. Birgitta is here. Thank you. Thank you very much. Wonderful. Now, it is my pleasure to invite our chair of our academic senate, Professor Amani Nuru Jeter, to the stage to tell us more about today's speaker. Amani, thank you.
Good afternoon. It is such a pleasure and honor to be here for this amazing, rich experience of the Faculty Research Lecture. Thank you, Chancellor Lyons, and greetings to all of our guests, and especially our former faculty research lecturers. The two individuals chosen to give this year's faculty research lectures are Henry Brady, Professor of Public Policy, who delivered his lecture on February 3rd, and today's distinguished speaker, Carlos Bustamante. Carlos is a professor of molecular and cell biology, physics, and chemistry, and a biophysics senior faculty scientist at Lawrence Berkeley National Laboratory. Professor Bustamante is interested in understanding the dynamics of molecular motors and the mechanisms by which they convert chemical energy into mechanical work through the development of force and displacement. In particular, his laboratory is investigating the physical basis of transcription regulation of RNA polymerase II by the nucleosomal barrier and the mechanism of translocation by the ribosome. His laboratory is also interested in establishing how proteins fold, which continues to be one of the most challenging questions in biophysics today. To this end, Bustamante and his team develop and apply novel methods of single molecule force and fluorescence spectroscopy, such as optical tweezers, magnetic tweezers, and fluorescence microscopy. Bustamante is a member of both the National Academy of Science and the American Academy of Arts and Sciences, and he is a Howard Hughes Medical Institute investigator. Now, I'll admit, as you could probably tell from my pronunciation, that I had to get AI to explain some of this stuff to me. But as a public health scientist who studies the social causes of population health inequities, including such things as how the social environment changes not our genes, but the expression of our genes, and how the social environment and social experiences contribute to health inequities through premature cellular aging, due to increased cell replication and telomere attrition, for example. Carlos's work has been and continues to contribute to foundational understandings to the mechanisms underlying these and many other biological processes, which is critical to the work that I and so many of my colleagues do in trying to better understand and seek to explain population health dynamics. Now more than ever, I would be remiss to not mention that it is imperative that we continue to speak these scientific truths. On behalf of the Academic Senate, Carlos, we are thrilled and honored to name you as one of our 2 faculty research lecturers this year. We are deeply humbled to have you amongst our colleagues. Everyone, please join me in welcoming Professor Carlos Bustamante for his lecture entitled The Development of Single Molecule Force Spectroscopy.
Thank you very much. Thank you. Thank you very much, Professor Neuro Jeter, and thank you very much, Chancellor Lyons. I'm going to start by, first showing you the full title of my talk, which you can see here. I know that you all pay your fees to come in here, and there are no refunds. Anyway, let me start asking the question, why force? It turns out that in biology, energy has been the concept that is more typically used in order to rationalize biological processes. And the reason is that energy is rooted in the powerful formalism of thermodynamics that tells us, for example, that energy is conserved. That's the first law. Or that an isolated system, when it undergoes a spontaneous process, always increases its entropy. Force, on the other hand, is a much more subtle and complex physical quantity, as I wrote here, and has not been used by biologists very much to describe their experiments. We all have a sense of what force is, the intuition that comes from the resistance that we get when we try to lift a weight, for example, right? And yet, this concept of force has led to a lot of confusion in the pre-modern era and continues to be a subject of a lot of epistemological discussions. For those of you interested in this subject, I recommend this fascinating monograph by Max Jammer, who was a professor at the Hebrew University in Jerusalem, and who describes beautifully the history of the concept of force and how it continues to be discussed. And yet, it turns out that force, the idea of force, has been used from the very beginning during the establishment of physical chemistry as a discipline. For example, van der Waals explained why different states of matter exist gas, liquid, or solid, in terms of the forces that the molecules felt or exerted among themselves. And Svante Arrhenius, a few years later, suggested that the rate of a chemical reaction is determined by the rate at which a strain and stress state of the molecule may actually be attained during its reaction in the reaction coordinate. Now, these ideas were very reasonable and, in fact, were theoretical ideas, and they were accepted because they made sense, but until recently, there was no way to directly test some of these ideas. In 1987, I was at the University of New Mexico, and I asked the question, how do molecules actually move through a gel during gel electrophoresis? And gel electrophoresis, by the way, is a technique that is used a lot in biochemistry, where you have molecules of DNA forced to go through a sieve, a gel sieve, which separates the molecules by size. And the way we force them is by actually applying an electric field to these molecules, which move to the positive electrode. And we wanted to know what happens to the molecules inside the gel. So we decided to actually mix some of the gel with molecules of DNA, and then we labeled them with a dye that made them fluorescent, put them between a slide and a cover slip, put the whole thing in the microscope, apply an electric field, and this is what we saw, what you see here. You see that actually both arms of the DNA extend towards the positive electrode, but by chance, always one of the two arms is longer than the other, experiences more force, and will pull the other around the corner, as you will see here, right? The upper one is pulling the other one around the corner, but notice how quickly the losing arm actually catches up, right? And so that is what really caught my attention, how elastic the molecule of DNA is. And that gave me the idea of asking the next question, which is, could we grab a single molecule of DNA by its end, one molecule by its end, and pull it and study its elasticity by determining how much force we have to apply in order to extend the molecule? And so first, I want to remind you how DNA looks like, right? This is the Watson-Crick structure. You have here the base pairs, which are stacked on top of each other like pancakes, A's in front of T's, G's in front of C's. And then you have two regions of phosphate here highlighted in red and blue and black and blue for each one of the strands. The dashed lines correspond to a full period of the molecule. It's a double strand, right? So we decided to do a physics 1 experiment, what we call the physics 1 experiment. What we did is we grabbed molecules of DNA and we attached the molecules at one end to a cover slip, and then at the other end of the molecule, we put a microscopic bead that was denser than water, so that the attraction of the gravitation of the Earth will actually pull them down. And we attach 1, 2, 3, 4 beads to increase the weight which they were being pulled. And then we measured the distance, the extension of the molecule. Essentially, that's what we did. And in the process of doing this experiment, what we were doing is repeating an experiment that had been done 3 centuries before by Robert Hooke, who actually studied the elasticity or the behavior of springs, and he discovered that if he actually doubled the weight, in other words, the force acting on the spring, the extension of the spring also doubled. So essentially, he showed, this is what is called Robert Hooke's law, that the extension is proportional to the force. And he expressed that in this book in Latin as Huc tensio sic vis, which means like the extension, so goes the force. The force is proportional to the extension. So we decided to look at the extension of the molecules under this condition, and you see that even though this was a very crude experiment, because we were using the gravitation of Earth on these little beads in order to extend the molecule, you see that already this experiment started to show us that maybe the DNA molecule is not a Hookean spring, in the sense that the force is to be actually increasing faster than the extension, and so it's not linear, as we say it in the jargon, right? In our jargon. So, and yes, it was a crude experiment, but since we were using the whole planet as part of our experiment, we at least could claim that we had designed the largest molecular biophysics experiment done to that date. The next year, we improve the experiment and we use magnetic beads to actually, also microscopic beads, to actually extend the molecule. And now you see that we could get a very, very nice data here and how nonlinear, how non-Hookean is DNA as a spring. If it were Hookean, then it would actually follow this dashed dotted line in blue here, but of course it is not. And so we published this in 1992, which I think can be considered like the date of birth of the single molecule force spectroscopy. Now notice that the units are in piconewtons, 5, 10, 15, 20 piconewtons. A newton is the weight of an apple, approximately and appropriately. An apple on the surface of the Earth, right, is 100 grams times, let's say, about 10 meters per second square acceleration. That's 1 newton. A piconewton is a millionth of a millionth of a newton. And so what this experiment showed was that it was possible indeed to mechanically manipulate molecules and apply forces that we could generate in the laboratory to study their behavior. So since we have this very nice data, we said, let's treat mathematically the data and try to see if we can explain or describe it mathematically. And in fact, in collaboration with Eric Silla at Cornell and John Marco, we developed this expression here in which we treated the molecule as if it could be made of a continuous elastic material, what is called a worm-like chain. And you see that the expression that we got here, the second term is the Hookean term, see, because the force is directly proportional to the extension. But the previous term, you see that actually the force is inversely proportional to the square of the extension, and that's the one that is responsible for DNA to behave so funnily, right? Not like a Hookean spring. Okay, so then we decided to test the theory with the experiment, and you can see that the theory that was being used up to that point by polymer physicists, which is the dashed line here, which is called the freely oriented chain, completely misses the data that we had obtained in our experiment, and that the continuous line, that is the new theory, the worm-like chain theory, perfectly fits the data. Now, as you know, in physics and also in biophysics, research is often limited by technology. And so when somebody invents an instrument that allows you to make a new kind of measurement, then a lot of interesting results may appear. And this is what happened when in 1970, Arthur Ashkin invented a technique, he was working at the time at Bell Labs, and he built an instrument in which he used a focused laser beam that could allow him to grab, push, trap, and move matter. He called this instrument optical tweezers, and in fact, because of this, for the discovery of the optical tweezers, he was awarded in 2018 the Nobel Prize in Physics. So, optical tweezers are fantastic instruments. We built them in our laboratory, and we've been perfecting them every year, because with them, you can actually exert force on objects, and not only that, determine how much force you are exerting on the objects, and also determine how the objects deform under the force or move places under the force. And if the objects themselves are able to generate force, then you can actually determine how much force these objects are generated. So we use optical tweezers, build the optical tweezers in the lab to study individual molecules. And now we can do experiments such as this. We can place a single molecule of DNA between a bead atop a pipette and another bead small bead that is held in an optical tweezer or optical trap, and then you can actually see that it is possible now to manipulate the top bead and extend it. And when we do that, we are measuring how much force we need to generate in order to extend the molecule for a given amount. So the experiment got much better now, right? Anyway, so let me now change gears for a second, because I'd like to show you that, in fact, there are many processes in biology in which force and not energy is the quantity that rationalizes the behavior of the biological system. And the way I'm going to show you, but before I do that, I want to remind you that in molecular biology, we recognize certain principles that have been found to be true for almost all systems, as special, and in this case, one particular one is called the central dogma of molecular biology. The central dogma of molecular biology simply says that DNA, the information of DNA, is used to make RNA, and the information of RNA is used to make proteins. Now, the process by which the information of RNA is used for proteins is called, here is, by the way, this is a molecule of RNA as we would describe it normally. It's a sequence of bases A, C, G, U, etc., right? And in the sequence, there is information, the genetic information, because the RNA is a facsimile copy of DNA. And now, the process by which you go from RNA to proteins is called translation, and the translation is carried out by the largest machine in the cell, which is called the ribosome. Okay, now, the ribosome reads the information in RNA in triplets or codons. So, for example, in the RNA that I just showed you before, you have this sequence, and so the first three are used to encode one amino acid, which is the language of proteins. Proteins are polymers of amino acids. So for every 3 letters in the RNA, the ribosome places one amino acid. The next 3 letters, another amino acid, and so on and so forth. So the result is that you end up with a protein. I'm calling it protein 1. But suppose that instead of starting at the first letter, the ribosome starts at the second base. So here you have a situation where you start here now, not in the first A, but in the second A. So now the first triplet is no longer AAC, but it is now ACG, right? Which is, I'm showing you here. And the next one is 3As, which is a different amino acid. So look, you got a different protein with the same RNA, simply by reading it in a slightly different frame. Okay, so the same thing, of course, is the ribosome starts reading a base earlier. The process is called frame shifting. And it was found in the 1970s that many viruses maximize the information content of their RNA, of their genome, by inducing frame shifting in the cells, in the host cells. And now you say, how did they do that? And the way they do it is because the RNAs of these viruses form certain structures that are very organized and are called pseudonodes. And these pseudonodes apparently are very difficult to disrupt. And so when the ribosome gets to the pseudonode, it stops and it cannot proceed. And then it starts moving back and forth. And in this process of moving back and forth, it frame shifts. Okay? So this is the idea. And pseudonodes then were discovered not only in viruses, but they were discovered also even in human cells. We know that actually pseudonodes play a role. And what was discovered is that the efficiency with which a pseudonode could induce frame shifting changed from pseudonode to pseudonode. And so the immediate explanation that it was proposed was that probably the pseudonodes that were more efficient at inducing frame shifting were the ones that were energetically more stable. So they tested the idea, and they found that that was not the case. It is not the energy what actually explains the efficiency of the pseudonodes. And so, at the time, Nacho Tinoco and I decided to actually test whether if instead of actually being the energy, it is maybe the mechanical stability, not the energetic stability, what actually determines the efficiency of pseudonodes. Before I tell you, and I show you the experiment that we did, I want to tell you a little anecdote because Nacho Tinoco, who was my research advisor in the 70s when these pseudonodes and the virus doing this were discovered, decided that actually he was going to do a contest in the laboratory with all his graduate students and postdocs who could write the best sentence made out of 3-letter words that will make sense if you start reading it in the first letter, the second letter, or the third letter, which is exactly what happens in biology because you read in the first letter, the second letter, the third, and you get proteins that work, and that work for the virus particularly. So the first prize was a 12-pack, the second prize was a 6-pack, and the third prize was a handshake by Nacho. I decided to enter the contest, and you guessed correctly, I only got the handshake. So in any case, we did this experiment, and so Nacho and I decided to do this experiment in which we grab a pseudonode, we attach it to a bead in a pipette, and then we attach the other end to a bit that is in the optical tweezers, and then we essentially measure how much force was required to undo the structure of the pseudoknot. And what you will see in the next slide is essentially what we found. Here is a plot of the efficiency of the frame shifting, and here is the force that we had to apply in order to undo them. And you can see that there is a very good correlation between the force required to undo the pseudoknots and the efficiency. And so this is an example in which force rather than energy rationalizes. What it's telling us is that in many respects, biology is mechanical in some aspects of it, at least. But the mechanical paradigm is not new in biology. I want to read you here this quote that says, "The operative industry of nature is so prolific that machines will eventually be found not only unknown to us, but also unimaginable by our mind." So wrote in 1666 Marcello Malpighi in his book De Viscerum Structura, which is actually shown here. Marcello Malpighi was a professor at the University of Bologna, the first university in the Western world, and he is considered the father of microscopic anatomy because he was using the microscope to look at biological systems for the first time, and he came up with this realization. Now, Malpighi was the leader of the revolution that swept through the biological sciences in the 17th century, and which paralleled the revolution that was occurring in the physical sciences at the same time, when Galileo Galilei first and Isaac Newton afterwards perfected the concepts of inertia, mass, acceleration that are the basis of our description of kinematics and dynamics, and which incidentally is the language in which you would want to describe the behavior of these machines that Malpighi was talking about. It's interesting to think that in both revolutions, the biological revolution and the physical revolution, it was the invention of instruments that allow scientists to see beyond the human eye, namely the microscope in biology, the telescope in physics, that ultimately were the catalyst that brought in both disciplines the modern scientific imagination. And machines, yes. There has always been some fascination of humans with machines throughout the centuries. Maybe it is because of the effects or the transformative power that some simple devices, as shown here, have, in which an action can be actually changed in direction or be amplified or be made to act in a different place. And this may be the basis of this enthronement. In any case, the word machine comes from the Ionian Greek word mechane, which essentially describes a contraption, some kind of mechanism. And the Greek term most likely comes from the Hebrew mechonot. Mechonot is a word that appears in the Torah to describe four-wheeled cars that were used by Solomon for the temple. The word in Latin is machina, as shown here, and already in the 3rd century, Archimedes was able to classify these machines as levers, pulleys, or screws, and even he discovered the mechanical advantage of the lever. So scientists have been interested in machines for a long time, and in fact, we know now that to perform directional movements, the cell uses tiny devices that operate as machines, and some of them, in fact, operate as molecular motors. We call them molecular motors because just like the engine in our car, they are able to convert the chemical energy of the fuel they use into mechanical work. And you remember that work in mechanics is simply force times displacement. And so these machines should be able to generate force and produce displacement, right? Now, the cell has thousands of different molecular motors we know now that, as Malpighi predicted, carry out many of the functions of the cell. And you can immediately see that actually because of the way these devices operate, single molecule force spectroscopy is a very good way to actually study these machines. So for example, imagine that you are looking or trying to study a motor, a molecular motor, which I depict here as this little guy in red, who is walking on this track, and if we attach this guy by some linker to a bead that is kept in an optical tweezers or optical track, then we can learn a lot about this motor. For example, how big are its steps when it actually takes one step? How big are the steps, right? Or we can say, what is the maximum force against which the motor can work? Because since we are grabbing the motor by the back, so to speak, we can always pull it back and see how much force it can generate until it stops. Or can it shift gears like our cars? Can this machine shift gears? Or how is this velocity affected when you increase the force against which the machine is working? And other questions a little bit more specific, like how tight is the coupling between chemical energy and force, or what is the mode of operation, whether it is a ratchet or a power stroke motor. So I'm going to give you two examples of machines that we have studied in the laboratory. The first one is one that you have already seen, that I already mentioned, and is the ribosome, which reads the RNA and produces a protein out of reading the RNA, right? And so in this case, we are giving the RNA is forming this structure that we call an RNA hairpin. Now, the ribosome is down here and sitting at the base of the RNA. We are grabbing the ends of the RNA, both strands of the RNA, and attaching them to one bead in a pipette and the other bead in the optical tweezers or trap. Now, if the ribosome wants to translate, that's translation, the process, right? Translate this RNA, it has to invade the RNA and it has to open the RNA. And so what will happen is that the RNA is like this, and as the ribosome invades the RNA, it will start doing this, right? And therefore, the distance between the beads, this bead and that bead, will tend to increase. And so we decided to study this motor, and this is what we observed. Remember that I told you that the ribosome always reads the RNA in triplets, 3 bases, right? So here is what we obtained. Oops, sorry. I hope this is real time, translation in real time by one single ribosome. And you see that actually it's moving exactly by 3 bases as it actually progresses in time. And so doing this type of experiments, for example, you can learn a lot of things about the ribosome. Of course, we already knew that it moved by 3 bases. But what we didn't know is that if you change, for example, the mechanical stability of the hairpin, the ribosome changes gear. Changes gear in order to be more efficient, in order to actually cross. Just like we do when our car, we find a hill, and then we have to actually change gear in order to accomplish the task. Here is a nicer and more beautiful description of this experiment. And you can see the enormous complexity of the ribosome, right? And also notice that in this case, what we actually were grabbing was the RNA and not the ribosome itself. We are not applying any force to the ribosome. We are just grabbing the RNAs. But we may want to apply force to the ribosome if we want to learn, for example, how powerful it is. And so we want to, in that case, play tug of war with a single ribosome, right? And so here is what we did here. In this case, we grabbed the ribosome, attached it by its back to the bead in the pipette, and then the other part, the distal part of the RNA was attached to the bead in the trap. And we could apply force, increase or decrease this force. Now, the ribosome is attached to a bead that is on a pipette, so it cannot move itself. So in order to translate the RNA, the ribosome has to do this, right? It has to thread the messenger RNA through itself. Motion is relative, right? Movement is relative. But in the process of doing that, it has to actually work against the force with which you are applying with the optical trap. And here is what we saw in this case. Oh. This is a much harder experiment because you really are measuring 3 bases of RNA, which is very elastic. And it still is possible to see that the ribosome is very, very precisely moving by 3 base pairs. The only imprecision is the quality of our data, which has noise. But the ribosome knows exactly what it's doing, right? Now, it turns out that you can actually repeat this experiment increasing the force more and more against which the ribosome is working, and you can determine how much force the ribosome can ultimately generate. And here is the answer. You can see here the velocity of the ribosome as a function of the force load that you place on the ribosome. And you can see that it decreases very dramatically, and eventually by about 12 piconewtons, the ribosome can no longer translate. So the ribosome is not so strong, but the cell wants it that way. The cell doesn't want something that will actually just go like a bulldozer through any barrier. It wants to actually stop and then regulate whether the ribosome can pass or not by using other factors. The second story that I'm going to tell you about is about a virus that infects bacteria, which is called a bacteriophage. That's the language we give to these things. And this one in particular is FIT29. This virus actually has, like all viruses, a viral capsid. And at the base of the capsid, there is this device, which is the packaging motor. And so the virus uses this motor to grab the DNA, its own DNA, and burn ATP, which is the energy currency of the cell, to start putting the DNA inside the capsid during the assembly of the virus. Now, this method of putting the genome inside the capsid is used by other viruses, many of them human pathogens, like, for example, the herpes virus, the polio virus, the adenoma virus. They all use a packaging motor to put their genome inside the capsid. Now, in this particular case, the motor is made by this pentamer. You see here is this pentameric structure, which is, we call these subunits, which are really ATPases that have the ability of hydrolyze or burn ATP, if you want, and convert the energy of ATP into force. And there are 5 of them, and they are all identical. 5 identical ATPases. So we could actually do an experiment like this, where we grab one single virus, and we grab the DNA that has not yet been packaged, the distal part of the DNA, attach it to a bead, which is in an optical trap, and then follow the packaging by a single virus. Okay, and so let me just quickly show you this. And you can see that this is in real time. There is the actual experiment, and you will see that in packaging the DNA, the virus has to essentially pull the bead out of the center of the trap, which is what is happening here. But it's hard to see but we can actually determine what's happening, because on the black line that is here on the right, you see that this is plotting the force against which the virus is working. Each tick mark here is 20 piconewtons. So it's already working against 40 piconewtons. 40 piconewtons, and it continues packaging. Notice it's going to 50 piconewtons pretty soon, and at one point the tether breaks and the bead will go back into the center of the trap, so that you believe me that indeed the virus is pulling there. They're pulling the bead out of the trap during packaging. Now, you can use these experiments and plot instead, you can plot the tether length, which obviously with time will get shorter and shorter because the bead is being pulled as a function of time. Because this is distance versus time, this is what we call a molecular trajectory. This is the terminology that physicists use for distance versus time. And the slope, the initial slope of this trajectory is, of course, the rate of packaging at the beginning of the virus. And these are 4 different viruses. We just put them one next to each other. And that initial slope is 100 base pairs per second. So this virus can package the DNA at about 100 base pairs per second. Pretty good, actually. But notice that the slope, actually, as the packaging proceeds, starts getting smaller. It's not the initial value of the slope, which is 100 base pairs per second. Now here, after packaging is occurring, the slope is actually getting smaller, as if there is a resistance that is working against the packaging of the DNA. So this is a very powerful motor, and here is a histogram of the stall forces that we determine for this motor. The average stall force is 55 piconewtons. But there are viruses that we observe that were working as high as 70 piconewtons or even a little bit more, right? And you may ask the question, why? Is it 70 piconewtons, 50 piconewtons strong? Well, myosin in our muscle, which is the main motor in our muscles, can only generate between 3 and 5 piconewtons of force. So this guy is 25 times or so stronger than myosin in our muscles. And you say, why? Why would nature go through the trouble of evolving a powerful motor like this? And the answer is that we determined that at the end of packaging, in the capsid of the virus, actually, the pressure of DNA in the capsid is 30 atmospheres of pressure, or 3 megapascals for the physicists in the audience. So 30 atmospheres. Now, to again gauge our sense of what 30 atmospheres are, let me remind you that when you open a bottle of champagne, or cava, or spumante, internationally, this wine can only be sold as such if it has dissolved 5 atmospheres of carbon dioxide in the wine. So when you open the bottle of champagne, the cork comes out at 5 atmospheres. The DNA at the end of packaging in the virus is at 6 times the pressure of a cork in the bottle of champagne. Okay? Now, so the next time you open a bottle of champagne, think of the virus, okay? Because it's a fit. Now, but then you say, still, why actually go through the trouble when all nature had to do was to make the capsid bigger? And then it wouldn't have to generate so much pressure, such a strong motor. The reason is that after the cell actually has made, after the host cell has allowed the virus to reproduce inside, the cell breaks and the new viruses go out to infect other cells. And they sit on the surface of the cell. They are in the extracellular medium. There is no ATP here. In fact, the tail has been put to the capsid and the motor has been removed. So there is no motor anymore. And the question is, how do I inject my DNA in the new cell? And so the solution that nature has found is once again mechanical. It has converted the chemical energy of ATP, burning ATP, right, ATP hydrolysis, into potential energy in the form of a loaded spring of DNA at 30 atmospheres of pressure. And when the time comes, it uses that 30 atmospheres to inject the DNA inside the bacterium. So you see again that actually this is one example in which, again, the mechanical aspect of biology actually becomes evident. Okay, so let me now tell you about the fact that we continue studying this molecule, this virus, and we develop even a nicer optical tweezers, the super resolution optical tweezers, both temporally and spatial resolution. It used 2 optical traps, so we could actually see things that we couldn't see with the old system. And there you are, there is the DNA, the capsid, and then we add ATP, and immediately we start seeing, and we can follow the packaging of the DNA getting shorter, but we have enough resolution to see that this trajectory is made out, it's like a staircase, made out of periods in which there is no packaging and then periods of packaging, and no packaging and packaging, like that, right? So, in fact, here is the molecular trajectories. There is a period where there is no packaging. This is the length of the DNA. There is no change in the length. We call them dwells. And then there is 10 base pairs package here. We call them burst, 10 base pair package, and then dwell, 10 base package, etc., right? Notice that I plot here many different curves because some of them are obtained at very little ATP, when we put very little ATP, and here where we put a lot of ATP. And the size of the burst, which is the dashed line, the distance between the dashed lines, is always 10 base pairs, regardless. The only thing that changes when we go from little ATP to a lot of ATP is that the dwell times become very, very short. So that immediately tells us that the binding of the fuel, ATP, to the motor happens during the dwell. When you put very little, the motor is waiting and waiting until finally it binds all the ATP that it needs, 5 of them. And when you put a lot of ATP, the motor binds them very quickly, right? And so the dwells are short. Okay, we have enough resolution to actually see how is one of these 10 base pairs bursts made. And I told you that there are 5 ATPases, right? 5 subunits. And there are 10 base pairs that are being packaged. So if I ask you how many base pairs are packaged by each subunit, you say it's 10 divided by 5, 2 base pairs. And that's what we expected to see. But then my student comes to my office and says, okay, I got the result. It is not 10 base pairs, it's 2.5 base pairs. So it's not 5 steps of 2 base pairs, but 4 steps of 2.5 base pairs. And I say, what? Impossible. You know that there are 5 subunits, and what you're telling me is that one of the subunits is not doing anything. Impossible. And they are all identical, the subunits, right? So I said, no, you have to repeat the experiment. We'll never publish this because besides, biology don't like non-integer numbers. So we better find 2 base pairs. So he goes back, he actually repeats the experiment and comes back and says, you were right, Carlos. It's not 2.5 base pairs. Actually, it's 2.4 base pairs plus minus 0.1 base pairs. So he had actually obtained an exquisite data with very, very little noise. And that really meant that out of the 5 subunits, somehow there is a symmetry breaking in the system that actually made one of the subunits to act differently, or at least not to work, right? So I call it the special subunits. My students call it the principal investigator, of course. And in any case, I want to add you one additional piece of information, and is that we also showed that the motor makes electrostatic contact with the DNA phosphates. Remember, the DNA has these regions of phosphates. Every 10 phosphates, so every 10 phosphates, it has to grab a phosphate, and every 10 phosphates, and so on and so forth. It doesn't do it with any phosphate, only with the Crick strand, not with the Watson strand. And we could demonstrate that. And also that these contacts were both important as load-bearing contacts and also had a regulatory function. So here is what we call the operational cycle of the motor, which in analogy to combustion engine, we can describe it in terms of a sequence of chemical and mechanical events. In red are the things that are depicted here in red, and in green is what happens that are depicted here in green. So fuel intake, the binding of ATP, happens during the duel, I told you, and in fact, exhaust of products of combustion, namely ADP, happens also during the red in an interlaced fashion, right? So then the packaging, of course, coincides with the release of phosphate during the burst, and you have 2.5, 2.5, 2.5, 2.5, 4 times 2.5, and that's the packaging. Okay, so now the question, of course, is what breaks the symmetry of the motor? What event breaks the symmetry of the motor? What events determine the identity of the special subunit under normal operation conditions? We found the answer to this question when we were trying to answer a different question, as sometimes happens in science, and is that we were asking, does the motor rotate the DNA while it packages it? We know that the motor can generate force. What we are asking now is, can the motor actually generate torque, right? And so in order to answer that question, what we did is we grabbed, again, a bacteriophage head here, or capsid, and we have the DNA attached to a bead in a pipette, and we put a tiny little bead on the side of the DNA. And then we added ATP, and this is what we saw. So in fact, what this really is telling us is that the motor not only can generate force, but it also rotates the DNA and can generate torque. And when we determined how much it rotates the DNA, we found that it was 1.4 degrees per base pair, or 14 degrees for 10 base pairs. And that gave us the clue of the model that we have now to explain what is happening and what makes a special subunit special. I am almost at the end of my talk, but I'm going to present you the model. The model is this. Let's assume that the special subunit is the one that contacts the DNA phosphate every 10 base pairs. So it retains its identity as the master regulator of the whole cycle. But it has to do it cycle after cycle after cycle. Now, the motor packages the DNA in increment of 10 base pairs. I show you, right? The bursts are 10 base pairs. If the periodicity of DNA were 10 base pairs, then after packaging 10 base pairs, the special subunit will immediately encounter the phosphate that it needs to contact in order to maintain or retain its identity. that the periodicity of DNA is not 10 base pairs, it's 10.4 base pairs, which means that after pulling the DNA by 10 base pairs, the phosphate that the special subunit wants to contact is not where it needs to be, but is 14 degrees away. And so the motor has to rotate the DNA by 14 degrees every 10 base pairs in order to contact the phosphate and retain its identity as being the master regulator of the cycle. So here is the penultimate slide where we have the current model of how the motor works. At the beginning of the cycle, the special subunit binds the phosphate after rotating the DNA, and that leads to the release of ADP. That allows one ATP to bind, which produces the release of the next ADP in the next subunit, which allows the next ATP to bind, release, bind, release, bind, release, until all of the subunits have bound ATP. And now the special subunit is the first one, we also showed that, the first one to hydrolyze is ATP, and tell the first mechanical subunits, now work, right? You have to do the work. And so the first subunit hydrolyzes ATP and translocate 2.5. The next one hydrolyzes, translocate, etc. At the end of the cycle, the phosphate is 14 degrees away. The motor has to rotate the DNA in order to grab the phosphate again to start the cycle again. So let me show you a little video here that more or less depicts how we understand the motor works. At the beginning, at the end of the previous cycle, the motor is full of ADP. And now the special subunit exchanges ATP and then it produces ATP. The binding of ATP produces the leaving of ADP, binding of ATP, leaving of ADP, binding of ATP, leaving of ATP. Now everything has ATP. The special subunit hydrolyzes and tells the first subunit, translocate 2.5, 2.5, 2.5, 2.5. Where is my phosphate? The DNA gets rotated by 14 degrees, and that leads to the release of ADP, and the cycle starts again. So this is how we actually understand now this machine. And I have two final thoughts to actually tell you about. The first is that, as you can see, viruses, despite their simplicity, are extremely sophisticated. So obviously, we can never underestimate them, right? And we know that. The second is that I hope that in this lecture I have at least made some of the case that actually biology is more mechanical than we have given it credit for. Forces are everywhere in biology and they play important roles. Finally, I want to thank my wife, Sylvia, who is here, whose love, help, and support has made my work possible. I also want to thank my children, Fernanda, who is here, and Carlos Jr., who is not here, for their enormous patience across the years, for all the time that I spent in the laboratory. Finally, I want to thank also my colleagues here at Cal. It was my dream to actually come back to Berkeley after being a graduate student and be a faculty member, and their collegiality and help and support really has been invaluable for my work. Thank you all, and let me show you the working subunits as opposed to the principal investigator. Here are some of my ex-graduate students and postdocs, and also some collaborators among the faculty. Thank you very much.
That was a mind-blowing lecture. I'm a cognitive scientist, and I came here thinking I wouldn't understand the damn thing because there's such a different level of abstraction. And surely I did hear you talk about machines and mechanical, chemical motors and stuff, but you also said, I wrote it down, the cell makes a decision not to do this. The ribosome knows exactly what it's doing. The cell wants it that way. It just dies when the structure makes sense. Are you doing cognitive science at the molecular level? Seriously, you use those words.
Yes, yes. They can easily be misunderstood, misinterpreted. But let me clarify. Thank you for the question. Well, this is the result of eons of evolution. Things that don't work the way they end up working actually are discarded. And so in biology, the systems have actually moved through trial and error until they find solutions that actually work. And in the cell, it's very important for the cell to be able to control the processes, not just the processes to occur like in a chemical reaction where you mix A and B and it's going to give us C. The cell will not allow that to happen. It has to be controlled. Control because it wants to, well, that's the living state. In my opinion, and in the opinion of my colleagues, the most interesting state of matter, living state. The living state is different because it's matter with a purpose to maintain its organization. Maintain its organization.
It makes sense. It's making decisions. The structures make sense.
Yes.
Thank you very much. You're now a doctor scientist.
Yes.
Does the need for rotation have anything to do with the chemical structure of the DNA, or are they just one have nothing to do with it?
No, yes, thank you for the question. So does the rotation have anything to do with the periodicity of the DNA? Oh, yes. The question is, does the rotation that you observe of 14 degrees have to do with the periodicity of the DNA, the helical structure of the DNA? And the answer is yes, because, of course, the periodicity of DNA repeats itself, at least structurally, if not in sequence, repeats itself every 10.4 base pairs. And so the motor packages the DNA by almost a whole period of the DNA, but not quite. And because it is not exactly 10.4 what it packages, but 10, it ends up actually far away from the next phosphate that the special subunit needs to actually contact. And so, in fact, the answer is yes. And I can tell you one more thing. We actually have done experiments and published experiments in which we put a piece of RNA in the middle of the DNA. Now, RNA also forms a double helical structure, but the periodicity now, instead of being 10.4 base pairs, is 9.1 base pairs. And then we ask, does the burst change? And it changes. The motor is actually trying to adapt itself to the periodicity of the molecule that is packaging. And so it's quite surprising that actually, and that makes sense, let me just say, because it may look like actually I'm talking about metaphysics here. I'm not, this is all evolved matter. But what I'm trying to say is this, that actually the periodicity of DNA changes with the sequence of DNA. And so when you have a DNA that is actually made up of all kinds of A, T, G, C sequence, you cannot tell that it's exactly 10.4. It's 10.4 on average. But it varies from sequence to sequence as the periodicity is changing. The DNA is not a perfect crystal. And so the motor has evolved the ability to adapt to the size of the periodicity in order to be able to be efficient about packaging. Sorry, I'm sorry. Yes.
So if you look at the different thermoregulation strategies of organisms, does the stalling force have a wide range with the poikilothermic
You mean with temperature?
temperature variation in a cold-blooded creature, basically? So you have at a statistical level, the mean molecular velocity.
Yes. So, excuse me. So yes, look, all these processes that we see that actually are being carried out by molecular machines that know exactly what they want to do, is, however, the result of the fact that they, at some point, they have to actually cross a barrier. And that crossing of barriers, chemical barriers, actually happens thermally. Nobody actually pushes the system to actually cross the barrier. The barrier is there, and there is the whole, at room temperature, the system is vibrating, and it finally jumps the barrier. So if you increase the temperature of the system, obviously it is a lot easier to cross barriers because the probability of actually finding a fluctuation that will allow you to cross the barrier increases proportionally. Yes.
And I think we have to call that a meeting. Thank you very, very much. Absolutely delightful. Absolutely delightful. You know, there were just so many phrases in there where you hear the phrase, it's like there's a lot behind that phrase, right? Even at the end where you said, this is evolved matter. Oh, how much thinking has gone into that idea? Molecular motors, biology is mechanical, energy versus force as the lens for understanding our world. Lovely. Thank you. Even as an obvious non-expert, I'm thrilled by your ideas. A recording of the lecture will be available to view on the Faculty Research Lectures website in approximately 2 weeks. And if you missed Henry Brady's wonderful lecture last month, the video is available to view now. Thank you for attending. Fiat Lux. Thank you.